and what it means for these very special places that we, in some ways, we take for granted. I mean, wilderness areas, we see them all around, but wildernesses need water too. And so this is part of the story that's going to hopefully preserve those things in perpetuity. So with that, take it away, Aaron.
Is anybody in the front row okay if I take off my mask? Sorry, I got a little sick the other day, but I think I'm on the mend. But anyways, thank you for the introduction, Dave. I appreciate it. And so I will get started. So I'm Aaron Klarenbach, as Dave introduced, and I'll be talking to you about aquatic macroinvertebrate community structure across across spatial scales and major disturbance in Central Arizona Highlands. Double clicker thing here, I think. All right, so here's that didn't work. Okay, figuring this out, but we got it. All right, so roadmap for where we're going today. So I'm gonna give a small general introduction, covering wilderness, water, and aquatic macroinvertebrates. Then the rest of this talk will follow my general thesis structure. So we'll talk about aquatic macroinvertebrate communities across spatial scales. And then for chapter 2, talk about a flood response in the community, and then follow up with a quick summary. All right, so there's going to be 3 themes that tie together my talk today, as Dave introduced earlier. It's going to be wilderness, water, and aquatic macroinvertebrates. So to start off the talk, I'd just like to share a small passage from the Wilderness Act of 1964, the definition of wilderness. So wilderness, in contrast with those areas where man and his own works dominate the landscape, is hereby recognized as an area where the earth and its community of life are untrammeled by man, or man himself is a visitor who does not remain. And though I like this somewhat poetic definition of wilderness, the concept of wilderness has remained somewhat controversial since its inception. However, I'm not going to be discussing that in depth too much here. I will instead be discussing wilderness as a federally designated land type and how we might contribute to that continued protection. So why study wildernesses? Why is it important? So wildernesses act as an ecological refuge. They help protect ecosystems by minimizing human disturbance. They provide great scientific study locations for relatively intact natural systems. They hold value for indigenous peoples and local communities, and offer a place of solitude and connection with nature away from civilization. And they protect headwaters, which are critical water sources, particularly in the Western United States. However, in Central Arizona, where my study system is focused, the loss of water is a significant threat to these systems. Two of the main threats are climate change through droughts, reduced snowpack, earlier seasonal snowmelt, which leads to longer dry seasons between larger base flows. And also the consumption of groundwater, so mining, agriculture, and municipal requirements. So water is a limiting resource in these wildernesses in Central Arizona. It's the lifeblood of these systems. It shapes the canyons. It's been doing that for millennia. Provides habitat for many taxa, aquatic producers, invertebrates, fish, amphibians, birds, mammals. And it does this further by supporting aquatic and riparian vegetation, both of which contribute to providing more habitat and more food sources to all of those different taxa. However, even in designated wildernesses, water isn't protected by default. Base flows are decreasing. And so what does a wilderness protection even mean if stream beds go dry and life that relies on this water disappears. So I'm an ecologist, not a lawyer, so I'm not going to go too much into the details of this. But I want to touch on it because it's one of the few mechanisms through which we can secure minimum base flows for wilderness areas. So the Federal Reserve water right is a right that ensures enough water is available to support the purpose for which the land was reserved. In our case, in wildernesses, it's a minimum flow required to maintain wilderness character as specified in the Wilderness Act. And here, just a quick snapshot of a recent case in the Gila River adjudication for the Arapahoe Canyon Wilderness. This happened several years ago, but the key part to note for this was the case was very important for setting a precedent by affirming that wilderness areas are entitled to protection under federal law, but those flows must be clearly justified and quantified. So that's what our team has the purpose of setting out and doing. So those of us working on aquatic macroinvertebrates, Justin and myself, are just one part of a larger team consisting of scientists from lots of different disciplines. So we have hydrologists, botanists, we have people working with fish, amphibians, and birds. And the overall goal is to provide the scientific justification to support those flows for the Federal Reserve water rights. And specifically for us, we are going to study the aquatic macroinvertebrate communities and the habitats required to support them. So our big overarching management questions, where does aquatic macroinvertebrate food base diversity occur within and across wilderness streams, and how do flood events affect aquatic macroinvertebrate diversity and biomass in wilderness streams? So my study sites, as I mentioned, is in the Central Arizona Highlands. This is a very transitional zone. There's a very large elevational gradient across my sites. So up to the northeast, you have the Colorado Plateau and the Mogollon Rim, and down to the southwest, you have it bordered by the Basin Range lowlands down there and deserts. The area is semi-arid, receives about 60 centimeters of annual precipitation over the course of the year, and these systems are interesting because they're both snowmelt driven and experience seasonal monsoons. So one of my sites is the West Clear Creek Wilderness. The West Clear Creek flows through here. The next one is the Wet Beaver Wilderness with Wet Beaver Creek, followed by Red Rock Secret Mountain Wilderness, and the creek that we're working on here is the West Fork of the Oak Creek. And last but not least is the Sycamore Canyon Wilderness with Sycamore Creek, though that shouldn't be confused with the other famous Sycamore Creek in Arizona. So I'm going to show you all a quick little video here that might better showcase some of the work that we do. So it's fine. Oh. So here's some work that we're doing in one of the wildernesses.
Aaron.
So people taking fish samples or fish counts. Working with lots of hydrologists. We're going to get ARU data at one point, but that didn't work out. Got some point birds. I've worked in some pretty cool places. And worked with a lot of really cool people as well. The video, I don't know if any of you all have heard of them, but Freshwater's Illustrated, they're the ones who made this video, and there will probably be more to come in the future. But yeah, these are just a lot of the other agencies that we work with. If sound is playing, it might be awkward for any other videos, but we'll see how that goes. Cool. So, all right. So, why study aquatic macroinvertebrates? So they're ubiquitous. They're found in almost all freshwater ecosystems. They're pretty easy to sample and relatively easy to identify. So caveat there. Benthic macroinvertebrates serve as indicators of stream health. Yeah, they have diverse life histories, and they can reflect a variety of impacts to the system, such as chemical contamination or eutrophication. Specifically, the abundance of specific taxa can indicate the health of a system. For example, if there's an abundance of stoneflies, mayflies, and caddisflies, so those are EPT taxa, then we know that the system is probably healthy, since those taxa are sensitive to degraded habitats. Additionally, they're a critical part of the riparian food web, linking aquatic and terrestrial zones, and serving as food for terrestrial consumers. So here's a kind of a food web from Baxter et al. We can see aquatic macroinvertebrates. They eat both aquatic primary production and molluscous material that falls from leaves and other riparian veg, and they contribute to the food web, supporting fish, lizards, birds, bats, and spiders. Here's some cool pictures that I took. Here's a canyon tree frog. It's really cute. And over here, I thought this was really cool. I got to observe this. Damselfly was emerging, and the spider just came and nabbed it at its most horrible time before it could emerge into a beautiful damselfly. But yes. So my first chapter, we're going to be looking at these different spatial scales. So the questions that I'm going to cover here are going to be: how do spatial scale and local environmental variables influence aquatic macroinvertebrate community structure? Question 2, how does aquatic macroinvertebrate richness, diversity, and density differ at the mesohabitat scale? And 3, how does aquatic insect emergence differ between mesohabitats? So first, we have to talk about this spatial scale thing that I mentioned. So each spatial scale is associated with specific environmental factors, and together these spatial scales act as like nested filters, influencing the species assemblages that we find there. So stream networks, they have this kind of nested hierarchical spatial structure. And while the stream spatial scale is quite broad, and it has a broad array of environmental conditions such as temperature, flow regime, and so on, the cumulative effects of all of these different variables can influence the community structure that we find there. So our streams are isolated in space pretty far. They're also canyons, so this might influence dispersal ability of the aquatic macroinvertebrates. So this could be playing a role in how the communities are structured. Next, we have the reach spatial scale. So a reach is just defined as a relatively short section of a stream or river, about a few hundred meters long, that is treated as a single unit for study or for management. And at this spatial scale, the big difference that we are expecting here is that reaches very high from the drainage can differ significantly to those closer to the headwaters, or sorry, to those to the confluence, to things like temperature and substrate size, and so on. And finally, the last spatial scale that we'll be looking at is the mesohabitat spatial scale. So these are ripple and pool sequences. So these are two types of mesohabitats that occur in repeating sequences within stream reaches. And riffles are stream sections that are shallow and fast flowing, while pools are these deeper and very slow, almost still waters that we see in streams. So at the top here, we have an example of a pool. This is one of our sites at Wood Beaver Creek, and here's a riffle, with riffle sites at Wood Beaver Creek as well. And we define the pools and riffles to have these characteristics when we were sampling them. So our sampling scheme, we had 4 different streams. So those 4 different streams within the wildernesses that I mentioned, we had 2 reaches on each stream. So upper and lower reaches, we had 2 mesohabitats within each reach, so riffles and pools, and then within each riffle and pool, we had 3 sample replicates. So we had a total of 48 samples. So our methodology, we collected samples using 500 micron DNet with an effort limited fixed area sampling protocol. We processed the invertebrates, stored them in ethanol for transport back to here, where we could process them. Spent a lot of time at the microscope counting and finding invertebrates, IDing them, and finally uploading them into a huge spreadsheet with about 1,400 rows and 35 columns to be worked on in R. So back to that first question. So how does spatial scale influence aquatic macroinvertebrate community structure? So we have this figure here. So this shows spatial scale, the different spatial scales that I talked about on the x-axis, and mean dissimilarity on the y-axis. So we essentially started with species by site matrix. We did a little fancy math to calculate the pairwise dissimilarities between sites. Then we took the mean for each spatial scale comparison. We controlled for other spatial scales by only comparing sites differing at that specific spatial scale being compared. So for example, we took the mean dissimilarity for pools compared to riffles, but only within the same reach and the same stream to isolate those other spatial scale factors. And so we see here that the most diversity is introduced at the mesohabitat scale. So that's about 0.75 there for mesohabitat, followed by stream and then reach. So for this figure, this is an MDS ordination. So for those of you who are not familiar with it, this figure is showing sites in species space. And so all you really need to know for this is that the closer the points are together, the more similar the communities are to each other. And so we see for here on the top one, we have riffles in red and pools on blue on the left. The riffle communities and the pool communities are isolated from each other. They differ significantly. There's no overlap. And as well as the riffle communities are very tightly clustered. So they're very similar to each other and have a bit less dispersion than the pool group does. The pool communities are a little bit more distinct from each other than riffle communities are from each other. For the next spatial scale, we have reach. So the reach spatial scale tells a little bit of a different story. They do overlap in species space. However, the upper reaches have a much larger dispersion compared to the lower reaches. This probably indicates that the upper reaches are much more isolated in space from each other, as well as with network connectivity. And so those may be driving the factors that we see there. And finally, we have the stream spatial scale. These spatial scales are, you can see the different colors aligned with the different wildernesses on the map. And we have Sycamore Creek falling out significantly from the other sites. And this is likely due to Sycamore Creek being the warmest of the 4 wildernesses that we had. And yes. Okay, so our question 1 takeaways. So how does spatial scale influence aquatic macroinvertebrate community structure? So we saw that mesohabitat is a scale at which most diversity is introduced in the system. So the highest dissimilarity was found there. And each spatial scale influences community structure. However, the strongest differences in community composition occur at the mesohabitat scale. So we know now that mesohabitat scale is the most important. So we can dive a little bit more into that. So how does aquatic macroinvertebrate richness and density differ at the mesohabitat scale? We will get into that. So to answer this question. Anyways,
We try to fix that. Sorry, getting so used to it, I'm not seeing it anymore.
But so to help answer that question, so we conducted an indicator species analysis. So this identifies how strongly a species is associated with specific habitat. So there's two components that go into an indicator species. So it's fidelity. How often does it show up in that specific mesohabitat, and then exclusivity, essentially. Does it avoid other habitats? And high indicator value means that a species is both common and restricted to that habitat. And so we might expect that taxa with high indicator values might be at risk if that habitat were to be lost. So we got, don't want to overwhelm anyone with tables, but we got a lot of indicator taxa. This is a very high diversity of taxa that we found in our system. So we have 31 indicator taxa within riffles and 7 indicator taxa within pools. So we see that riffles, they have this very distinct and specialized community with all these taxa here, and this matches the structure that we saw with the NMDS ordination. So if you remember, the riffle ordination was a lot tighter than the pool ordination in species space. So a lot more of these taxa are occurring at more of those sites, as opposed to the pool sites, which are very dispersed in species space, meaning that some of these taxa are not showing up at all the different sites, except for some of the ones shown here. So we can see from this list, for example, that the loss of ripple habitats through perhaps decreased stream flows would disproportionately affect these taxa, because there's so many of them that are only found there. So we found that taxonomic richness was significantly higher in riffles and pools. This might be because of the high habitat heterogeneity that we find in riffles. There's a lot of larger substrates that influence interstitial spaces, as well as a lot of variable water velocity. So there's a lot of different niches that organisms can occupy within riffle habitat. We found that density is significantly higher in riffles than in pools as well. And this is probably due to riffles being highly oxygenated. They're very productive habitats. They're stable substrate for colonization compared to a pool, which has very fine sediments that can easily be washed away. As well as there may be lower predation pressure in riffles. There's a lot more places to hide between different rocks and interstitial spaces within riffles. And we also found a higher proportion of individuals with complex life stages in riffles than pools. So what do we mean by this? So we mean taxa that will transition to an adult stage that is terrestrially available, that makes them available to terrestrial consumers. So lizards, birds, bats, frogs, and so on. And so we see much higher numbers in riffles. And again, this may be because taxa like snails and clams and so on might be a little bit more, have a reduced predation pressure from fish, for example. Within pools, they can just kind of do their own thing, because crunching on a snail shell probably it's not very appetizing. So the question two takeaways. So how does aquatic macroinvertebrate richness and density differ at the mesohabitat level? Aquatic macroinvertebrate richness, diversity, density, and number of indicator taxa, and the proportion of individuals who had complex life cycles were highest within riffle mesohabitats. And since riffles are typically the first to dry, riffle communities are the most at risk. And so how does aquatic insect emergence differ between mesohabitats? We know that these riffles are hotspots in the wilderness streams. So do we see the same pattern with emergence? So just a little background on how we did this. We did two streams, Sycamore and West Cliffs Creek. We did two mesohabitats, riffle and pool habitats, and we had 4 traps per mesohabitat, and then deployed these traps over a 4-day period. And this is what we found. We did find that they have a significantly different community structure. However, there's a bit of overlap in pools and riffles. We did not see any difference in emergent biomass or EPT biomass between riffles and pools, unfortunately. I guess that was just the case. But this might be due to several reasons. One, it might be the low sample size. And 2, it may be that emergent taxa don't show specificity for emergence location. So our question 3 takeaways: how does aquatic insect emergence differ between mesohabitats? Emergent aquatic insect communities differ significantly between riffle and pools. So maintaining both riffle and pool mesohabitats would support a diverse emergence of aquatic insects, supporting higher trophic levels. I'm going to go to chapter 2 here. So in this chapter, we are going to look at how the aquatic macroinvertebrates respond to a huge flood that we saw in the system. So the questions that we hope to cover or answer for this is: how do aquatic macroinvertebrate communities respond to a large flood? And then how do population responses compare with the theoretical models of disturbance recovery? And so why study this? Floods are expected to increase in magnitude and frequency, and the effects of extremely large naturally occurring floods are pretty difficult to study. They usually don't get a lot of advance notice on when a huge flood is occurring, so you don't get to get out there and sample beforehand. And so we had a great opportunity to do this because we had collected samples in Sycamore Canyon before a massive flood. And so this is a wonderful opportunity for us to do kind of like a little natural history observation of how communities respond. So we're going to be focusing on Sycamore Canyon Wilderness. So this is the 2nd largest canyon in Arizona. It's the largest of the 4 wildernesses. And it's a huge area. It's 3 national forests, the Kaibab, Prescott, and Sylvania National Forests, are all within its boundaries, or part of it falls within their boundaries rather. So on to the flood. So we saw this huge storm on August 18th, 2021. That dropped a large amount of rainfall in the early afternoon and evening. About 4 to 6 inches fell in this area of the Mogollon Rim. And here highlighted in this white, it says where it says Raphael. There's a Raphael, that's a Raphael burn scar from a wildfire that occurred just a few months earlier in June that year. That'll play a part in later in the talk. But so that huge amount of rainfall, the 4 to 6 inches, that fell traveled through the burn scar and down through the various canyons, particularly through Sycamore Canyon, Sycamore Creek, before it converted with the Verde River. And unfortunately, there's no USGS gauge on Sycamore Creek prior to this flood. So we had to rely on what the Verde River gauge showed us at Clarkdale near the confluence. And we saw a huge rise in the stream gauge. We registered about a 900 cubic meter flood, or a 32,000 cubic foot per second flood. So this was a massive flood for this system. I don't really have a way to put it into scale for you, but the recurrence interval of this flood was estimated to be about a 500-year flood. So you wouldn't see another one of these for a really, really, really long time in this canyon. So when we got there in fall of 2021 to sample our aquatic macroinvertebrates again, this is one of the scenes that we were greeted with. So we saw a huge amount of woody debris that probably ended up here because all of these trees were burned from that wildfire that burned earlier in the year. And so this is very destructive. A lot of woody debris ended up in side channels along the stream bank and so on. We saw a lot of woody debris deposited up in the trees up here and along the different on the canyon walls and so on. So it's a very destructive event. And so yeah, how did the aquatic macroinvertebrates fare through all of this? But for our methods, very similar to what we did for the other project, but we sampled for 4 different time series. Once we had 1 sample from before the flood and 3 times post flood. And then we also did biomass using published length weight regressions. So for biomass and density, we did not see a difference pre and post flood here. There was a small difference between fall 22 and spring of 22, but that is not really what we're focused on here. So why do we see no change there? Question 1, we saw we did see a difference, in richness and diversity between pre and post flood. Here, so we see that richness and diversity drops down post-flood and then slowly rises back to pre-flood levels. So this is an interesting pattern when you combine it with what we saw with the biomass density, how they didn't change. So maybe the NMDS ordination will reveal something. So we do see that communities differ significantly on the NMDS ordination. So we have spring 21 in blue, so this is pre-flood. We have fall 21 in orange here, spring of 22 in this purple color, and fall 22 in yellow color. And so the pattern that I think pops out immediately for probably most people, as it did to me, was that of seasonality. So we see the spring samples grouped together as well as the fall sample kind of grouped together. So if there is a pattern here because of the flood, we might have some seasonality that's obscuring it. So, so what are the key takeaways here? Oops, sorry. How do aquatic macroinvertebrate communities respond following a large flood? So we did see that aquatic macroinvertebrate community structure differed in response to the flood. And we did see community richness and diversity decrease in response to the flood, but it recovered by fall of 2022, while community-wide density and biomass did not differ significantly. So we know that the community structure changes, but that may be due to seasonality. We also saw an interesting trend with richness and diversity dropping post-flood despite no change in biomass and density. So do we think that the underlying population level responses can help us make sense of the patterns that we see at the community level? So how do population response compare with theoretical models of disturbance recovery? So here's one of the, oops, I guess that's not a video. So this is one of the things that we saw when we came back in fall 21 was this huge explosion of blackfly larvae simulium all over the ripple sampling sites. There's just so many of them. It was kind of fascinating to see them in such huge numbers. And so this is kind of what prompted this. I guess that's just a short aside. But so what does disturbance theory predict? So we expect that past life cycle taxa are going to show rapid recovery with a post-flood boom. Slow life cycle taxa are expected to show delayed recovery, and then finally non-flood adapted taxa are expected to show a very, very delayed recovery. This is a figure from Lauren McMullen's paper showing population responses in that model. So what did we see empirically? So we saw a post-flood boom for fast life cycle taxa. So Simulium, the one that I showed you a picture of, that was the one that keyed us in. We see a huge rise in density in that first sampling season post-flood, followed by a sharp increase in biomass by spring of 2022 before dropping off there. We did another taxa, so Falcion, this is a baited mayfly. Another riffle specialist, we see a very similar pattern for this taxa as well. And we did one more baited mayfly, Calvatus. However, this one's a pool specialist. And we did see a boom in fall 21. However, that dropped off pretty quickly by spring 2022. So this kind of matches what we expected to see. So we see this huge pulse right after the flood for these fast life cycle taxa. So for the slow life cycle taxa, we did see a delayed recovery. So for Tetrophila, so this is an aquatic moth. This was lost from the system entirely until fall 2022, or at least we did not detect it. And this might be because these are Tetrophila depend on rooted macrophytes, which were probably scoured away during the flood. And another taxa that we saw this pattern was Chamara. This is a little black caddisfly, and we see that they also decreased following the flood, and then slowly started to return by fall of 2022. So these are univoltine, so they have one generation per year. So you can imagine if the flood wiped out most of the taxa, they experienced high mortality in the spring 21. There weren't a lot left by the spring 2022 to finish the life cycle. And additionally, they build their retreats on the undersides of rocks and riffles. So it's probably pretty likely that they experienced very high mortality because of that. And for our non-flood adaptive taxa, we have Ostracoda. They were pretty much devastated by the flood, not really returning to any pre-flood levels. They reproduce quickly, but they just don't really have a way to avoid floods or recolonize effectively once they're wiped out. And we have, I'm not trying to say that exactly, but Gyrolis, it's the ramsport snail, similar story as with the Ostracoda, they kind of fall off completely after the flood. Key takeaways, observed population responses to the flood aligned with responses predicted by theory. Fast life cycle taxa rapidly recovered and boomed above pre-flood levels, while slower life cycle taxa exhibited a much slower recovery. The overcompensatory boom by fast life cycle taxa is likely responsible for the lack of change in density or biomass at the community-wide level, despite the loss of a significant number of taxa post-flood. So, answering all those other questions, did we answer our management questions? So where does aquatic macroinvertebrate food-based diversity occur within and across wilderness streams? So while each spatial scale contributes to structuring the aquatic macroinvertebrate community, the mesohabitat scale adds the most diversity to the system, and riffles are exceptionally productive and contain a diverse community. And our other question, how did flood events affect aquatic macroinvertebrate diversity and biomass in wilderness streams? We saw that floods restructure habitats and reset successional stages, increasing diversity and overall diversity in the system. And with that, oops, I'd like to thank you all. Oh, this went too fast. Hold on. Anyways, so I'd like to thank my funding sources, Oregon State University and U.S. Forest Service for funding this project. I was very fortunate to be able to work on this and work with a lot of great people and go to a lot of cool places to do this work. I'd like to thank my committee. I look forward to chatting with you all in a little bit. Yeah, I hope you don't mind me using the pictures that I could find of the Oregon State website. Thanks to our lab, I wouldn't have been able to do any of this without you all. You all have been really important for helping me through all of this. Shout out to Angelica specifically. You have mentored me a lot throughout all of this, and I'm really grateful. I thank you to all my friends. Again, I don't know that I'd be here without you all supporting me along the way. And thank you to my family from supporting me from afar. And I don't know that I would be able to finish off this talk without thanking the wildernesses themselves. They provided me a lot, and I guess particularly a place of solace when I needed it the most. So, yeah. And with that, I'll take any questions.
Your NMDS plots, do you know what variables were most influencing your axes?
The environmental variables?
Yeah.
Yes. So I didn't really get into it because I didn't want to overly confuse anybody, but the NMDS1 org axis, the x-axis for the organizations was particularly driven, or was, sorry, strongly correlated with hydraulic variables. So water velocity and depth were strongly associated at that axis. So that clearly differentiates the pool versus ripple communities. That makes a lot of sense. And then the NMDS2 axis, the y-axis, that was very strongly correlated with elevation, temperature, and conductivity, all of which are related to temperature. So elevation and temperature are inversely proportional, and then conductivity increases proportionally with temperature with ionic mobility.
You mentioned flood adapted aquatic invertebrates, and I was curious if you knew of any, like what type of adaptations that they might have that might benefit them in a disturbance like a flood, besides just like, I don't know, reproducing quickly, if they had any.
Yeah.
That would benefit them.
So there's lots of different adaptations. One of Dave's favorite taxa has a very unique flood adapted trait. So giant water bugs, Avidas herberti, I don't know how to say it. So they sense a cue from heavy rainfalls, and when they sense this, they'll start crawling up the canyon walls to avoid the big floods that come through. Other taxa might avoid it just by being in their adult life stage during a flood. So they're in their adult stage, a flood comes through, they just have to mate, lay eggs, and then it's free real estate. Sorry, bad.
Awesome talk. With the flood, so there's a fire, like the burn scar in the flood area. Do you think that that had any impact on like the recovery of invertebrates downstream beyond just making it so the flood was larger, like less water? Like, anything about like the fire in particular that might have changed invertebrate response?
Yeah, I think that there's a ton there. I have not researched wildfires and floods, although there's a lot of research on that. I've not looked into it specifically, but I imagine, so one, you have tons of large woody debris entering the system, so that's a lot of increased habitat heterogeneity, so that's more places for aquatic invertebrates to live. You also have those nutrients that are entering the system. So that's further introducing a lot of different variables in the system. I'm imagining you have, with the wildfires, you have a lot of the substrate soils was washed away, so it's no longer supported, kept in place by those trees that were there. So yeah, I imagine there's a lot. I unfortunately don't know as much about that particular topic as I kind of should.
Great talk, great photos. On the NMDS that showed the pre and post flood, and it had that strong seasonality effect, there was also a difference in the spread between fall of 21 and fall of 22. Do you think that difference is potentially because of the flood, or is it hard to say that that could be attributed?
Yeah, I think that's very hard to say. I think if you remember the density and biomass plot, I know I was going very quick at the end, but we saw that biomass and density were both significantly lower than all the other ones. I'm not exactly sure why that was, although I suspect there was a small flood pulse that went through about a month before we took those last samples, and so that was like very shortly before that, but I'm not sure why that difference between the two fall sampling seasons might have been. The flood could be a very good reason, but it's hard to know definitively, I think.
Obviously, the flood in your study was tremendously large, but they have monsoons yearly.
Mhm.
So how common is it to have minor floods? And what is or is there variation in floods, or is it like every 10 years you get a bigger flood, or is it,
Yeah, so yes,
pattern?
there's there's a lot of floods that come through here. I mean, I think I was looking at the hydrograph. I think again, it was last year or 2 years ago, there was another exceptionally large flood that finally showed up on the Sycamore gauge that they just installed after this flood. And that was, I think, almost a 20,000 cubic foot per second flood. So just another monster roaring through there. I think Dave was actually there at the tail end of that as it was receding. And so it experiences a lot of flood variability.
That was the only time in my life I've been at a stream and didn't find a single invertebrate. They were gone. It was in a section that is normally dry too, so not much, but it was a shocker.
Does that answer your question?
Okay, well, let's take Aaron one more time. Fun is just beginning.